Well, welcome to this podcast on the Nurse Education on Podcast series. Today we're going to be looking at diabetes mellitus, but before we do that we need to look at some background anatomy and physiology. And of course when we're talking about diabetes and blood sugar control, the place we need to start at is the pancreas. And if you've got one of the white physiology books, you might find it helpful to open it to page 155. And if you've got one of the black pathophysiology books, you might find it helpful to open that to page 266. Now, the pancreas is described as an organ with a head, a body, and a tail. It's located in the upper part of the abdomen, so it's below the diaphragm, and it's behind the stomach. It's a fairly posterior organ. And the head is towards the right-hand side, and the tail is towards the left-hand side. And the pancreas itself sits in the curve of the duodenum. Now, the interesting thing about the pancreas is it's got two different sets of functions, because it's an exocrine gland and it's an endocrine gland as well. Now, you might remember that an exocrine gland is, well, first of all, a gland is something that produces something. A gland produces a product. And an exocrine gland will secrete its product via ducts. So the product will be produced in the gland, go along a duct, and be secreted somewhere else. For example, sweat is produced in exocrine glands. It is produced in the gland, the sweat goes up the duct, and it's deposited on the surface of the skin. Saliva is produced in exocrine glands. There are salivary ducts draining the saliva from the gland where the saliva is produced into the mouth. They are exocrine. And endocrine glands are glands which secrete their product directly into the bloodstream. They have no ducts. And I've got an old-fashioned physiology book in my office that was published in 1940, and it calls the endocrine system the ductless gland system. Glands without ducts. The products go directly into the bloodstream. And in the pancreas, most of the tissue in the pancreas is exocrine. It's actually producing digestive enzymes. And these digestive enzymes drain in ducts into the pancreatic duct through the ampulla and are deposited in the duodenum via through the sphincter of Oddi, the exocrine products. But we're not particularly interested in those today. We're interested in the endocrine products. Now, in the pancreas there's about 1 million little islands of tissue. And these were discovered by a guy called Langerhans, who named them after himself. So they used to be called the Islets of Langerhans. The more modern name is just the pancreatic islets. But there's about 1 million of these in any individual pancreas. And the interesting thing about these is that round about the outside they've got some cells called alpha cells. And in the middle they've got some cells called beta cells. Now the alpha cells produce an endocrine hormone called glucagon, and the beta cells produce an endocrine hormone called insulin. And the really clever thing about these cells is that the beta cells detect when blood sugar levels are increasing. They actually detect it. The cells themselves detect an increase in blood sugar levels. And it's when they detect the increase in blood sugar levels that they release the insulin. And the alpha cells detect drops in blood sugar levels. They detect hypoglycemias. So when the blood sugar level drops, the alpha cells respond by producing a hormone called glucagon. So if you eat a lot and there's lots of sugar in your blood, if you eat carbohydrates or sugars, they'll be broken down in the gut, they'll be absorbed. It doesn't matter what sugar what type of sugar is absorbed through the lining of the gut, because everything that's absorbed through the wall of the gut drains to the liver in the hepatic portal vein. And if there's any other form of sugar enters the liver that is not glucose, the liver will convert it fairly promptly into glucose. So the only sugar that you find in the systemic circulation is glucose. So glucose and blood sugar are interchangeable terms. Blood sugar is always glucose. So after eating, the levels of this are going to rise. That's going to be detected by the beta cells. The beta cells are going to respond by producing insulin. And insulin will lower blood sugar levels. You can't be any form of clinician if you don't realize that insulin lowers blood sugar levels. And let's look at a couple of ways that it does this. First of all, insulin will convert the soluble glucose molecules into insoluble molecules of glycogen. Now, glycogen is a starch, really. It's sometimes described as animal starch. It's a polysaccharide. It contains many sugar units. So insulin will link up hundreds and thousands of individual glucose molecules, forming them into huge molecules of glycogen. And once this glycogen is formed, it's stored in the liver and it's stored in the skeletal muscles. But can you see that if the insulin is converted from glucose to glycogen, the glucose is no longer in the blood? The glucose is now stored as insoluble glycogen. So because it's no longer in the blood sugar levels have dropped. Insulin is lowering blood sugar levels. But as well as that, insulin facilitates the movement of glucose from blood and tissue fluids into the cells of the body. Now, some cells of the body, particularly muscle cells, liver cells as well, glucose can't get into the cells easily or readily. And if you think about it, you might work out a reason for this. The reason is that cell membranes are made up of phospholipids. They are phospholipid bilayers. And that means, of course, that they're fatty. And glucose is a water-soluble molecule. Glucose dissolves readily in drinks. You put sugar in your tea, it dissolves readily. So glucose is a water-soluble molecule, but the cell membranes are made up of phospholipids. They are fatty. That means that glucose cannot diffuse into the fatty layer, because water-soluble molecules will not dissolve in fat. Glucose is described as a hydrophilic molecule. It's water-loving, but it's lipophobic. It fears fats. It cannot dissolve into fats. But of course, the mitochondria, which require the glucose, are inside the cytosol of each individual cell. Because remember, the whole point of getting the glucose is to combine the glucose with oxygen to produce energy. And as you might remember, in that equation of respiration, glucose plus oxygen gives energy, with the waste products of, or the byproducts of water and carbon dioxide. So we've got to get the glucose into the cell. So what actually happens? An insulin molecule will bind with a protein insulin receptor on the surface of the individual cells. Now, these insulin receptors are proteins, and they're suspended in the phospholipid bilayer. And the insulin molecule will bind to the external part of the insulin receptor. And insulin receptors are described as transmembrane molecules. They go all the way through the membrane. So the insulin molecule binds onto the receptor, and that triggers changes in the part of the insulin receptor which is inside the cytosol. And this triggers off what are called secondary messenger systems within the cell. And these secondary messenger systems activate molecules which are already in the cytosol, called GLUT molecules, G-L-U-T. GLU is glucose. They are glucose transporter molecules. And the way I think about these is they're like donuts with holes in the middle, or polo mints with holes in the middle. And these, under the influence of the secondary messenger system, after the insulin receptor has been stimulated by insulin, the secondary messenger system will cause these glucose transporter molecules to rise to the surface of the cell membrane, which means basically there is now a hole in the surface of the cell membrane, or a gate, and glucose will go through this gate. It's the only way that glucose can get into these cells. So glucose will go through the glucose transporter molecules and get into the cell. So that means all the components of this system must be intact for it to work. You must have the insulin to stimulate the insulin receptors. The insulin receptors must be healthy to receive the insulin and to facilitate the changes in the secondary messenger system. And the secondary messenger system must be working, and the glucose transporter molecules must be there to allow the glucose into the cell. It's interesting to note that potassium also nips in with the glucose. So when the glucose transporter molecules are on the surface of the cell, potassium can